So again, really grateful to have these two with us today. We have Dr. Ryan Thum, who received his bachelor's from Florida State University and his PhD from Dartmouth College. He's currently a research professor of genetics at Montana State University's Plant Sciences and Plant Pathology Department. His research focuses on the ecological genetics of invasive aquatic plants, with specific interest in identifying the underlying genetic architecture for herbicide response and developing genetic tools to help with management decisions. And he's joined today with Ashley Wolfe. So go ahead and take it away.
Great. Well, thanks all for being here, and I'm really honored to be invited to speak with you all, um, and share with you a great deal probably about our work on Eurasian watermilfoil and in particular integrating sort of genetic studies and herbicide studies with the intention of improving management outcomes. Excuse me. The co-author on the talk is Ashley Wolfe, who's on the screen there. So Ashley's a technician turned master's student in my lab. We've really put her through the ringer over the last few years, but she's really been an important driver and worker in a lot of the work that we're presenting today. So I'll do the presentation, but I think in the question and answer session, I anticipate there might be some questions that would be better directed at Ashley, or to Ashley, maybe I should say. Uh, Basically, the simple premise of the research program is that genetic variation can influence management outcomes. So we wouldn't expect every single plant of the same species to respond in exactly the same way to any given management implementation. And so our goal, sort of broad goal, is to use genetic information specifically to predict herbicide response and be able to improve management outcomes in that way. And so I like to draw the analogy. Many of you are probably familiar with like genetic health panels and personalized medicine. So you can get tested for a number of different things using something like 23andMe, or your doctor can order tests to see if you're a carrier of a known disease or if you have particular risk factors. So like this has been really, interesting for things like Alzheimer's and stuff like that. So um, same premise, not quite the same technology or same advancements yet, but this type of approach is very common in terrestrial agricultural weed management, but not so much in aquatic plant management. So in terrestrial weed management, it's pretty common to have either nucleic acid-based tests or protein-based tests that can fairly rapidly give you a good sense of whether or not the population you're dealing with is resistant versus susceptible to one or more potential alternative herbicides that you could use. And so we want to do the same thing in aquatic plant management. And then this is maybe a bit of a minor point, but I guess just something that I felt I should point out. Like this research program has both short and long-term approaches and objectives. And so I'm not going to really structure the talk by those, but just keep in mind like that, some of the things that we're doing in the long term will trump some of these things that we're doing in the shorter term. But we're not waiting for perfect conditions to make progress. So this will be a mix of sort of things that we're doing, both that have sort of more immediate impact versus our investments in longer term impact. A real quick introduction to the plant. If you're not familiar with it, this is the one of the plant species we work on, and certainly the plant species that we have the deepest, we've dug the deepest in the research program. So this is Eurasian watermilfoil, Myriophyllum spicatum. It's this very, in my opinion, beautiful, feathery leaved, submerged aquatic plant. Some of the sort of natural or life history that's really important for this talk. This plant, like many aquatic plants, reproduces both asexually and sexually. So really prolific vegetative reproducer, but still, like most aquatic plants, produces fertile flowers and retains the ability to reproduce sexually. And an important piece of our work is that the sexual reproduction and technically also what we call somatic mutation creates these distinct clones, which I might refer to as strains, that can propagate indefinitely through vegetative reproduction. So looking broadly across the landscape, we have a number of clonal lineages that can spread within and between lakes through clonal reproduction or vegetative reproduction. Importantly, this species, which is introduced, not native to North America, hybridizes with northern watermilfoil, Myriophyllum sibiricum, which just happens to be phylogenetically the sister species to Eurasian watermilfoil. So they're pretty distinct genetically, but they're each other's closest relatives. This figure here is just to illustrate genetic diversity in Eurasian and hybrid watermilfoil, which will be the focus of the talk today. I'll see if I can get my pointer to work. So this is a principal components analysis of about 2,000 molecular markers spread across the genome. Each point on this graph is just a unique genotype or a unique DNA fingerprint, if you will. Points closer to one another are more genetically similar than points more distant to one another. So here's a swath of genetic variation in our native northern watermilfoil. Here are two swaths of genetic variation in what we would consider pure Eurasian watermilfoil, but two distinct lineages. Um, probably representing distinct introductions, different introductions from different sources. And then both of those lineages have hybridized with northern watermilfoil. So we have a variety of distinct hybrid lineages or clones that have from this lineage of Eurasian hybridizing with northern, and another set of hybrids from representing hybridization events between this lineage of Eurasian and northern watermilfoil. So there's a lot of genetic diversity out there, but there's also a lot of clonal reproduction. So it's interesting in itself, I suppose, that there's molecular genetic variation or among individual variation in water milfoil. But what we're really interested in our lab is genetic variation for herbicide response. And so, short and sweet, there's a lot of genetic variation for herbicide response, and that'll be the focus of the talk today. Just really quickly to illustrate that for you. Here are a bunch of different strains tested for fluoridone, which is a PDS inhibiting systemic herbicide that's commonly used to control Eurasian water milfoil, and I'll talk quite a bit about that today. Each one of these panels, like I said, is a strain. On the y-axis is just the amount of biomass harvested at the end of the experiment. The left-hand dot is plants that are untreated. The right-hand dot are plants that have been treated with fluoridone. And so what you can see is there's a lot of different responses to fluoridone there. I'll point out one that's resistant here. So this one actually does better when you sort of quote unquote feed it fluoridone. And then here's one that's sensitive to fluoridone. And that's the typical response and what we would expect. This is another systemic herbicide. This is a systemic auxin, 2,4-D, two different strains of water milfoil. One happens to be a Eurasian water milfoil strain, the other a hybrid water milfoil strain. The x-axis is the concentration of 2,4-D. The y-axis is growth compared to untreated plants. What we see here is this strain has our sort of typical desired decrease or sensitivity to 2,4-D, whereas this strain is fairly resistant to especially lower concentrations of 2,4-D. So that genetic variation is what we're really interested in primarily in the research program, and I'm going to dig in a lot to that today. The way I'm going to organize the talk is at these different organizational levels. So we're talking about genetic variation that can be structured in a variety of different ways. So at the highest level, I'll talk about taxa. So that would be things distinguishing different species like Eurasian from northern water milfoil and their hybrids, as I mentioned, those two hybridize with one another. We'll focus in on strains or different clones. And that's because there's genetic variation for herbicide response even within these taxa. And we think, in our opinion and in our operation, that's sort of more the level that we focus in on. And then ideally, right, and then focusing in like at specific genes. So that's even sort of narrower than the strain level. So we're interested in identifying specific genes for herbicide response because different strains can share the same genes, just like many people in the human population share the same genetic risk factors, even though they're unrelated to one another. So looking at comparison between hybrid and Eurasian water milfoil, I'll give you the punchline here first, and then I'll walk through some figures to illustrate it. Hybrids on average do grow more vigorously than Eurasian water milfoil, and on average they're less sensitive to 2,4-D, especially at these lower concentrations. But there's some important nuance to that I'll get to after proving, I guess, this main point. So this slide sort of has two different studies that we've done over the past in the lab. The left-hand slide was work done by Liz LaRue, who's now a faculty member at University of Texas, El Paso. And what Liz did was she sampled a variety of hybrid and Eurasian water milfoil populations, primarily in Michigan, which is where we were at the time, and then kind of did a contrast or an overall comparison between hybrid response and Eurasian water milfoil response to various concentrations of 2,4-D. And so on the left-hand side, what you're looking at is a dose response curve that represents the contrast between those two taxa. Trying to get my pointer back. I've lost it, so oh well. So what you can see is at all levels of 2,4-D tested, we have better growth proportionally of hybrid water milfoil than Eurasian water milfoil. This is admittedly sort of fairly low concentrations of 2,4-D. I'll get back to that later, but these are operational concentrations. If you happen to be an aquatic plant person, target concentrations are often higher than this, but achieved concentrations are usually in this range. So that's kind of why we decided to focus there. Hannah Hoff is currently a PhD student at Brown University. She did her master's in the lab, and she extended Liz's work by taking that work down to the strain level. So this was an overall comparison of hybrid versus Eurasian. And then this is 4 different strains of Eurasian water milfoil in the black bars and 4 different strains of hybrid water milfoil in the gray bars. The left-hand panel here is untreated plants, and the right-hand panel here is plants treated at 500 parts per billion 2,4-D. The lines here, the dashed line is the grand mean for hybrids, and the solid line is the grand mean for the Eurasian water milfoil strains. And so what you can see in both untreated and treated plants, the hybrids are generally doing better than the Eurasian water milfoil. And I'll come back to that in a little bit, but there is an overall statistical effect of taxon when we're looking at things like growth in response to 2,4-D. This is a great study. This wasn't done by my lab, but I love pointing it out. This is work done by a lot of folks from the Wisconsin DNR, led by Michelle Nault. And so what they did was they compared the control efficacy in the field of Eurasian water milfoil lakes, so that's this top panel up here, compared to hybrid water milfoil lakes, this bottom panel down here. So they measured the 2,4-D concentration that they used, or that they achieved, I should say, and then they estimated percent control on the y-axis. And so what you can see is that generally very high control for pure Eurasian water milfoil in the 80 to 100% range, and comparatively lower percent control for hybrid water milfoil, so more down in the 50% range. And so a really nice field study just sort of illustrating this or demonstrating this taxonomic effect of hybrids versus Eurasian. This is a table that I lifted from an EPA label. This is a table of recommended doses for a newer oxinic herbicide, fluorpyroxyfenbenzyl. You may know it as Procelicor if you're familiar with aquatic herbicides. And so here are the recommendations for pure Eurasian water milfoil versus hybrid water milfoil. Generally, they have higher dose recommendations for hybrid water milfoil compared to Eurasian, and presumably that's predicated on the notion that hybrids tend to be more vigorous and tend to have lower sensitivity to auxins than pure Eurasian water milfoil. So distinguishing hybrid from Eurasian has a lot of meaning, and it's very easy to do. These are figures, again, lifted from work that's not necessarily my own. This is a genetic assay we've been using in my lab for a really long time, and then this was published by a group out of Ontario a few years ago. And so the upshot from it is it's very easy with sort of normal molecular lab benchtop kinds of apparatuses that you could distinguish Eurasian from hybrid water milfoil by amplifying a particular gene region known as the internal transcribe spacer, digesting that with a couple of different restriction enzymes, and then the different banding patterns that you get, which you can do on your benchtop, would tell you whether or not you have hybrid water milfoil, Eurasian, or northern water milfoil. So this is a service that my lab routinely does, and again, it has meaning because there is an overall effect of hybrid versus Eurasian water milfoil. So coming back to the punchline, hybrids on average grow more vigorously and are less sensitive to 2,4-D. That's especially true at lower concentrations, but that's really relevant. I'll touch on that a little bit more in the future here in the talk. That may also apply to other herbicides, especially at low concentrations, but we don't really have good data for that, so I won't say that that's necessarily true, although it's probably likely true. But the important caveat here is that this really varies by strain. And so it is not true that all hybrids are resistant to all herbicides. That is just absolutely not true, but that's a logic error that we can make if we take this inference too far. And it's also not true that all hybrids are more resistant than all Eurasian water milfoil. So it's not that black and white. I'll go back to the studies that I presented earlier. So from Hannah's work, right, we see an overall average better growth when untreated or treated with 2,4-D for hybrids, but we do have some strains of your pure Eurasian water milfoil that grow better than some strains of hybrid water milfoil. Okay, and so really the devil is in the details for this. It's really probably more the strain level that matters. If we look again at these field data from Wisconsin, yeah, most of the hybrid populations had on average lower efficacy or lower percent control than the Eurasian water milfoil, but there are also hybrid populations that are responding as well as Eurasian water milfoil. So more than likely, we don't have data from this on this particular study, but more than likely that reflects a difference in the actual strain composition of hybrid water milfoil in these lakes versus these lakes. Looking at fluoridone again, real quickly. So again, these are data that I had showed earlier, so I'll move fairly quickly through them. Here are two strains that are highly resistant to fluoridone. This strain is a hybrid water milfoil strain. This strain is a Eurasian water milfoil strain. So it's not like Eurasian water milfoil is immune from resistance and that it's only in hybrids. And then the flip side of that coin, here are several hybrid strains that are plenty sensitive to fluoridone. And so transitioning now, we really focus the research program not so much on comparing hybrid and Eurasian, although that's a meaningful comparison. We really think the more appropriate taxonomic unit operationally for us is the strain. And so this is where Ashley's work has really come in. Ashley's really taken the lead on characterizing water milfoil strains, and she's built this really excellent, well, I mean, maybe we're biased, but really cool app called Milfoil Mapper. So here's the link there. You can also find it on our website, or you can email me and we can get you a copy of it, or get you the link. So Ashley developed this app with Alex Badges at the Minnesota Aquatic Invasive Species Research Center. And it's a really slick app. And so I'll walk you through sort of how it works really quickly, and also like what we think people can do with it and what we hope people will do with it. So this is just a screenshot. This is the opening screenshot. When you open the map, those are just all the locations that we have water milfoil strain information for in the US. In the app, you can query essentially the underlying database for it. So you can explore by state or county, or if you know a particular lake, you can search for that lake and find it in the app. If you know something, if you have a strain that you're interested in that you know the name of, you can also browse by that. And this is an example of doing that. So this is when we queried for this strain that we just call hybrid 249. These are the locations where we've seen that strain before. So Milfoil Mapper will give you a map of all the locations where a particular strain has been found. It can print that list for you if you want. And then if you click on any particular lake, it will give you all of the strains that are known to occur in that lake. And it'll also tell you something about those strains if we know it. So in this particular lake, there's two strains there. One strain we don't really know much about in terms of herbicide response, but in terms of this hybrid 249 strain, we categorize that as sensitive to 2,4-D, but resistant to fluoridone. And I'll come back to those labels a little bit more. But so you could use that information in guiding your management decisions. So here's a very simple flow chart for how we envision people using this app. So, once you have a list of, once you know what strain or strains you've got in your lake or your collection of lakes, probably the most important thing that you'd want to know is whether or not we know anything about the herbicide response. In cases where we do, then we would argue that you want to take that information into careful consideration when you're designing your management tactics and your management prescriptions. And so obviously you'd want to avoid an herbicide that one or more of the strains is known to be resistant to, and you'd want to focus your management efforts on those herbicides that you know or that you have strong evidence that the strain is susceptible to. But even in the case where we don't have herbicide response information for strains, which admittedly is most of the strains at the moment, there's a variety of things that people could do. So you could talk to other lakes that have the same strain. So you could share experiences, what's worked, what hasn't worked. You can consider doing quantitative genetic monitoring. I'm not going to get into that in the talk today, but We have a few papers on that, and can certainly consult with people on whether it makes sense to do that, and if so, the logistics of doing that. And then another thing that I think people could creatively pool their resources together, right? So if there's a collection of lakes that all share the same strain, you could pool resources together potentially and do actually controlled comparisons in the field or in the lab. I'll give you a few examples of strains that fall into these two different categories. So a couple of strains that we know quite a bit about. This is Eurasian strain 734. It's very widespread. This is by far the most common strain of watermilfoil that we see across the country. And we have it categorized as sensitive to both 2,4-D and fluoridone. That's based on laboratory testing that we've done. In contrast, this is a strain, this hybrid 249, which I showed before. We have it categorized as sensitive to 2,4-D, but resistant to fluoridone. So this is really pretty highly resistant to fluoridone. It's commonly found in Michigan, so we recommend that Michiganders, if they're going to use fluoridone, which there's a good reason to many times, to screen their population for the presence of this particular strain, because it is found quite commonly. And really fluoridone's not going to work. So that's an example of sort of both sides of the coin, where here's a strain that we're not that concerned about for herbicide resistance, another strain that we are really concerned about for a particular herbicide. Few examples of strains that we don't have herbicide response information for, but maybe would want to prioritize to do that. Got one, we're in the Midwest here. Well, we're not physically located in the Midwest, but this is the Midwest Invasive Plant Network, and most of our work is done in the Midwest. So here's a few examples, but one each from Michigan, Wisconsin, and Minnesota. So here's a strain that's found in a handful of locations in Michigan, a strain that's found in a handful of locations in Wisconsin, and a strain that's found in a handful of locations in Minnesota. And we don't currently have these characterized, but it might be beneficial for lake associations or managers to know where the strain is also found, so that they can put their heads together and decide a best course of action. So a couple of important so where we're really at currently is identifying strains, mapping their geographic distributions, how common and widespread they are, and characterizing them for herbicide response. I wanted to take a few minutes and go through a few slides to really point out some nuance that I think is really important for this. And so the first is that resistance is situational. And if you're a terrestrial weed scientist, that may make you cringe a little bit, but I'm going to stick to that. And the reason I say resistance is situational, even though there's a very clear definition for what herbicide resistance is, it's still situational because, it's not a binary thing. It's not like a plant is either resistant or susceptible. That's really context dependent. And so resistance versus susceptibility is not an on-off switch. Instead, the underlying reality for plants is that they have what's called a dose response curve, so that they have some curve that maps out what the effect of an herbicide is given a particular dose. And that's a curve. It's not a flat line, and so that's why it's not really binary. That's really important, and probably particularly important for aquatic plant management compared to something like traditional terrestrial row crop agriculture, because the use patterns for aquatic herbicides, meaning the concentrations used and the exposure time that plants experience, really varies from place to place. Sometimes that's regulational, like there's regulations that are different state to state. Sometimes it's based on the management goals, and sometimes it's just plain logistics, like hydrological factors and things that are out of folks' control. And so there's a curve. I put all these dose response curves up here to just really make the point that there's a sort of underlying curve for all of their responses. But why that's important is, for example, this strain here, when used at 6 parts per billion fluoridone, we may expect a pretty good response by that strain. But if used at 3 parts per billion, we may not get a very good response at all. And so in Michigan, where we've done most of our fluoridone work, the operational fluoridone concentration is 6 parts per billion. But in other places that use fluoridone, they try to go quite a bit lower than that. And so you can see that like a strain, pointer back in, a strain like this would probably be resistant to 3 parts per billion, meaning not very good control. potentially in a place like Wisconsin or Minnesota. Whereas if used in Michigan, the control might be okay. In contrast, a strain like this over here, even using it as low as 3 parts per billion, you're probably going to have really good control over that. So resistance is our use of the term resistance is somewhat, not even somewhat, it is contextual. Another important nuance is that and this is a little bit sort of trippy when talking about genetic variation, but generally there's more genetic variation at lower herbicide doses or lower rates than at higher rates. And so I'll tell you the practical implication of that first, and then I'll try to illustrate that a bit in a minute. But what that means is that we can expect more variation lake to lake in our results of using a particular herbicide tactic when they're used at lower rates. And so an astute person might say, well, then just don't ever use them at lower rates. I'll get into this a little bit later, but for aquatic plant management, it's really common to use herbicides at low rates because we have some real important challenges with selectivity. Removing selectively target plants without keeping non-target plants down to a minimum. And so one of the ways to do that is by using these kind of lower rates. And so as a practical matter for aquatic plant management, which is, again, very different from terrestrial weed management, oftentimes we're using these low rates. And so down here is just an illustration. This goes back to the late 1980s when really 2,4-D was probably the only tool for Eurasian watermilfoil management, or at least the most common tool. It's still a common tool. But this shows the relationship between the exposure time of an herbicide and the concentration of an herbicide. And so generally at higher concentrations of an herbicide, you don't need as long of exposure times to have injury or control or death of that plant or that population. Whereas at lower concentrations, you need higher exposure times to have the same effect on that population. And so I'll just kind of illustrate the point in text there above by sort of reference to this figure. So here's a dose response. Here are dose response curves for 5 different Eurasian watermilfoil strains. And so when we're in this zone of high concentration and high exposure time, so that would be a high dose, we have very little genetic variation for herbicide response, right? So we see there's a little bit of variation here, but hardly any at all because most plants are killed by that really, really high dose. But when we're operating on the edge of this, which we just most commonly are as just an empirical reality, we see a lot more variation. So here, 500 parts per billion or half part per million exposure for 3 days, right? That's this piece of these curves, and we can see that there's a ton of variation in herbicide response there. So if we're going to understand variation in our operational herbicide management outcomes for Eurasian watermilfoil with any herbicide, but in this case 2,4-D, then we really have to understand the strain-specific responses, or at least the range of variation across strains to really get at that. I think in the interest of time, I'll do this really quickly, but as an important strategy moving forward, this is just a pitch I like to make, especially if there's any state agency folks in the audience. Like, if you're doing quantitative studies to sort of find that sweet spot of where you want to use a particular herbicide for a particular target plant, it's really important to not just go in the backyard, grab a population of that plant and assume that they're all going to respond similarly or the same. It's really important to sample the range of genetic diversity in that species and get some quantitative data that captures that range so that you're not being misled by the particular strain that you happen to have in the backyard. So that's just something I'd like to see the community do a better job of moving forward. Okay, so that's sort of the strain level, and that's really where we operate. But the problem with the challenge with thinking about strains as the operational taxonomic unit is that there's a lot of them. And so to have an exhaustive catalog of the response of every strain to every herbicide is, I mean, it's possible. It's within the realm of reality, but that's a very large undertaking. And so one way that we could kind of cheat on that is if we can find out what's going on at the genetic level in strains, then we can predict an uncharacterized strain's response to a given herbicide. And so that's the basic premise of things like personalized medicine and a lot of stuff even in terrestrial weed agriculture or um, terrestrial agriculture, weed management, or pest management in general. So real quickly, the rationale behind trying to identify or the goal or the rationale between wanting to identify the genetic basis to herbicide response is that, with our DNA fingerprinting, yes, we can distinguish different strains, we can characterize those strains, and we can distinguish resistant versus susceptible strains. And we have tools that do that now, and they do a decent job. But it's important to recognize that each of these strains can reproduce sexually. And when they do that, they pass on their resistance genes to new strains, and those new strains aren't characterized. So we'd have no way of really seeing that happen in the field. There's really no way to like assign a parent of a strain to a known characterized strain. And so if we can identify the specific genes, we can develop genetic tests for those genes, and then we can apply those to strains that aren't characterized. So for the last portion of this talk, that's what I'm going to focus on, and sort of let you know where we're at. Excuse me. So I'll give you an example of that. This is the only example that I'm aware of from aquatic plant management that actually has a genetic test for herbicide response. This comes from work done not by my lab, but done on hydrilla, which is probably on your radar in the Midwest. It's not nearly as common there as it would be in like the Atlantic or Gulf coastal states, but really, really troublesome plant commonly managed with fluridone. This figure here is a dose response curve for 4 different strains of hydrilla. The y-axis you can think of as growth, and the x-axis is the concentration of fluridone. So we have our wild type fluridone susceptible hydrilla, and then we have these 3 different strains that sort of exhibit this increasing degree of resistance to fluridone. We know very well the genetic basis to that resistance. So the difference in fluridone response by these 4 strains primarily is a function of uh, single base pair mutations at a very particular spot in a very specific gene. This is the phytoene desaturase gene, which produces the phytoene desaturase enzyme. That enzyme is the target of fluridone, and amino acid substitutions at the 304th codon position, 304th amino acid position in this enzyme leads to different levels of resistance. So depending upon the particular DNA substitution that happens there, you get these different levels of resistance. And so it's very easy to sequence that piece of DNA and then tell somebody or give a prediction on how fluridone would, a given level of fluridone would work on that strain or set of strains in a particular lake. So it's a really nice example of a genetic test for herbicide response. And we want to do the same thing in Eurasian watermilfoil and in other species as well. So I'll spend some time talking to you now about work that I'm really excited about. I mean, this is kind of like, probably 5 years ago, definitely 10 years ago. I mean, I would have never thought that the lab would be doing genomics and finding, sequencing whole genomes and doing the stuff that kind of still sort of blows my mind. So I'll try to contain my excitement while I go through this, and it might be, there's going to be some maybe weird looking figures and stuff, but um, I'll just give you kind of a tour of how our approach to finding genes for herbicide resistance. And this is all ongoing work. So I'll refer to these as genetic mapping populations. So genetic mapping is basically finding those spots in the genome that associate or cause a particular trait of an organism that we're interested in. So in this case, I'll be focusing on fluridone response. So what we did for this first few slides I'll show you, we crossed a known fluridone-resistant strain of watermilfoil to a known fluridone-susceptible strain. This is a strain isolated from Lansing Lake in Michigan. This is a hybrid strain isolated from Baseline Lake, also in Michigan. So we crossed those 2 together, which is fairly straightforward for us to do in the lab. And then we screen those products.
Okay. Ashley, are you seeing the pause here?
Yeah, I am.
Okay. Think- Oh, looks like we're back. Okay.
Okay, we're back. God, man, right when we get into the good part. Um, it says my internet connection is unstable, so I apologize if that's the case. Hopefully we'll get through this.
If you want to turn your video off, sometimes I know that helps. I would
Sure,
understand if that
do
might help
that.
things.
Yeah, you guys don't need to look at my ugly mug here. You want to look at the, let's see if I can figure out how to do that though. I have a banner that's blocking my, okay. So hopefully this will work okay now. So the left-hand side here, this is the susceptible parent from Baseline Lake. On the right-hand side, this is the resistant parent from Lake Lansing. And then these are four progeny or offspring plants from that, some of which are really susceptible, like the Lake Lansing parent. Oh, keep screwing that up. The Baseline parent, and some that are really resistant, like the Lansing parent. This is just a summary on the left here of all of the offspring. So we've got our resistant parent here on the left, untreated and treated. So you can see fluridone really doesn't affect that plant at all. And the susceptible parent on the right. And then this is the range of variation exhibited by the offspring from that cross. So one of the things that we can do then is we can statistically compare the genomes of those that are most similar to the resistant parent to those that are most similar to the susceptible parent. And that's kind of the way that this study works. So these are the results from that. I know this is a lot to look at. There's 21 figures here. That's one for each one of the 21 chromosomes that Eurasian watermilfoil has. So I'll dig into these a bit more in the next slide to kind of orient you to what they are. But all those blue dots are basically data points, and so there's many tens of thousands to hundreds of thousands of data points in these studies. This is zooming in just on chromosome 7. And so on the x-axis is the position along the chromosome. So starting from one end of the chromosome going all the way over to the other end. So this particular chromosome is like 40 million base pairs long. And then the y-axis is just the difference at each one of those spots in the set of resistant plants versus the susceptible plants. That might be a little abstract for non-geneticists, but basically we expect that value to be 0. And what we're looking for are areas of the genome where that value is unusually large. And so the green lines and the yellow lines are the confidence intervals. The blue dots are the individual data points. And the red line is like the rolling average of every 100 or so of these points. And so here's an example on chromosome 7 where we see some pretty compelling signal that there's unusually large difference between resistant plants and susceptible plants in these areas of this chromosome. We've identified a couple of chromosomes for which that's true for this population. This is chromosome 13, which also sort of flirts with um, being unusually different. We're probably more interested in this one, and I'll explain a little bit more about that in a minute. But we have a few promising leads of areas in the genome to explore that are associated with fluridone resistance in this particular strain of watermilfoil. And then interestingly, it does not appear to be related to the target site enzyme PDS because that's located on chromosome 10, 11, and 12 in this in Eurasian watermilfoil, and we don't have any signal in those regions. We constructed a second mapping population. So this is a hybrid watermilfoil strain isolated from Townline Lake in Michigan. This is that hybrid 249 strain that I showed you on Milfoil Mapper before. Done a lot of work on this strain. We selfed that strain and created a mapping population. And then this is looking at chromosome 7, again, from that mapping population. And here's the chromosome 7 from the previous population that I just showed you before. And what we can see is that we're seeing signal in that exact same spot of chromosome 7 here. And so that's really kind of boosts our confidence that there's something real going on there. And then by comparing these two, we can really sort of isolate the signal to sort of this region of the chromosome which is helpful. So there's evidence for the same gene region as in both of these populations, and it suggests that these two strains have the same or similar resistance mechanism, which would be really convenient for genetic tests if that turns out to be the case. But we're not certain of that yet. So that's kind of where we're at with the data analysis part, data collection analysis part of finding gene or genomic regions that are associated with resistance. I find that really very exciting as a geneticist. But then it's sort of like, okay, so what? Like, where do we go from there? So what we're working on now and into the future is developing and testing molecular markers that are associated with resistance. So we've identified these areas that we have compelling evidence are associated with resistance. Ideally, we would identify like the causal mutations for resistance and directly assay those. What I mean by that is I showed you the hydrilla data earlier, right? So we know the exact DNA substitutions or mutations that cause that resistance, and we'd like to identify those specific things. You can get away potentially without even knowing necessarily the specific mutations. You can use molecular markers that are indicators of resistance. So these would be things that are closely linked to the causal mutations, but maybe not the exact causal mutation, unless and until we know that. So that would be sort of designing molecular markers in these regions and testing them to see how much predictive power they have in for resistance. So we need to ultimately test that on uncharacterized strains. These would be strains that sort of occur out in nature that aren't in these mapping populations that we constructed. So we want to know whether or not those gene regions that we identify in our mapping populations translate broadly to the portfolio of strains out in the real world, or if they don't. And of course, that's going to depend very heavily on how many unique ways there are to be resistant. This is promising. It looks like these two unrelated strains have the same or very similar genetic mechanism. The type, we don't know that for sure, but it looks that way right now. And as I said before, that would be really convenient if that bears out to be true, because we'd have fewer genetic tests that we'd have to develop. So I'll end here, just try to kind of wrap up and summarize like what I think the benefit and relevance of this work is. And here I will divide it into the sort of short and long term. And so in the short term, what we're working on sort of the day-to-day basis right now is building this catalog of strain distribution and herbicide response information that then goes into Milfoil Mapper, this sort of public-facing, user interface that people can actually get their hands dirty with. That's important because when we identify strains that are resistant to particular strains or sensitive to particular strains, people can directly use that information. So I gave you the example of, I gave you a couple of examples of susceptible strains that are widespread and a resistant strain that's fairly widespread in Michigan. That part of the research program is also really important. We have to identify strains that are resistant versus susceptible in order to do the long-term work of genetic mapping. And then in the long term, this identification of the genetic basis of resistance will really facilitate the development of specific genetic tests like the ones that your doctor would order for you or prescribe for you, or 23andMe could give you. But these genetic tests for herbicide response that we can apply to strains that have not been characterized yet. And then sort of more broadly, this stuff is done pretty commonly in terrestrial agricultural weed management, and not so much in aquatic plant management, and also not so much in sort of terrestrial weeds of sort of natural lands and rangelands. And so, I hope that the work that we're doing is providing conceptual and methodological models for people to integrate genetic and genomic information into invasive plant management. There's a lot of people to thank. Ashley, in particular, who's an important contributor to all this work. A few other people that are really important to mention from the lab. Greg Chorak was a PhD postdoc who's now a postdoc at USDA. He's really kind of led the charge in the genomic and bioinformatics work. Del Haney is a current graduate student working on a lot of this stuff. These two gentlemen here are my sort of angel geniuses of bioinformatics that have really helped out in assembling genomes and helping us do genomic work in this non-model species, which is really non-trivial. This is Alex Harkis and Josh Clevenger from Hudson Alpha Institute. Ray Newman's at the University of Minnesota, and he's been a really important collaborator. A whole bunch of people that deserve mention, but there's not enough time or space on the slide um, to credit all of them. And then for funding, it's been primarily, the research program is primarily been funded by the U.S. Army Corps of Engineers Aquatic Plant Control Research Program. I've had a little bit of other federal funding from the Great Lakes Restoration Initiative and a variety of funding from various states and private entities as well. So I just want to acknowledge the sort of portfolio of agencies and entities that have helped facilitate this work. And with that, I will take a breath and shut up for a second and take any questions if anybody has any.
Well, thank you both so much for being here. This is really interesting, and I appreciate your time. And I see the applause is coming in the chat here, so I'm not alone. Um, I don't see any questions in the chat, but we can leave it for a minute and people, oh, someone just asked the question that I was going to pose. Um, so it said, is your labs accepting samples to identify strains? And I was going to sort of pose the same question. If you go to Milfoil Mapper and you don't see your lake information in there, is there a way to like request that?
Yes, and I will let Ashley take that one.
Yes, there is. So currently we don't have like dedicated funding to do that. So it would be fee for service. So it's kind of like if you don't see your lake in there and you're really curious to know what is in your lake in terms of watermilfoil strain composition, we can have like a little meeting. Obviously, I guess my email is on the website of Milfoil Mapper. So you could reach out to me or you could go to Thum Lab website and you can find mine and Ryan's email and reach out to both of us. But it starts out with like a meeting, kind of getting an idea of what you're hoping to find in the lake in terms of like what you're looking for, what information you want, and then we kind of go from there. But yes, currently we are still taking samples for this season. It would be kind of probably at this point knowing what's in your lake for future treatment for next season. But yeah.
Okay, excellent. And then I'll just, again, we can wait a few minutes if anyone still has a question, you can take a minute. Um, I was really interested, you were talking about the difference in like fluoridone response, specifically like how you had to use higher concentrations for a lot of the sites in Michigan, or not you, but that higher concentrations are needed there. Um, can you talk, I think you kind of addressed this, but can you talk a little bit more about like why resistance varies by location?
I think I'll probably take that one. Yeah, I mean, so the degree of resistance is a property of the plant itself. Um, the reason I say like sort of the use of the term is situational is that a state that uses fluoridone at 3 parts per billion is going to experience more like resistance than a population than a state that uses it at 6 parts per billion because there's going to be more genetic variation for fluoridone response at 3 parts per billion than there is at 6 parts per billion. And so, you know, Michigan just happens to use it at 6 parts per billion based on some studies that they did in the mid to late 1990s. And so when we're talking about fluoridone resistance in Michigan, we're sort of using the response at 6 parts per billion as our benchmark for whether or not fluoridone is going to give you acceptable control or not at that concentration. And what's different is, let's say, I mean, I don't want to speak for a state, so if there's anybody from Wisconsin or Michigan or Minnesota here that can correct me for this, but my understanding is that those states tend to use fluoridone less often. And when they do use it, they tend to use it at lower rates, so like 3 to 4 parts per billion. And so you may have a strain that if it was treated at 6 parts per billion would be fine, like it would work really well, but treated at 3 or 4 parts per billion, it wouldn't work as well. And so in that case, if the operational use rate is 3 to 4 parts per billion, then that strain would be considered resistant to that rate of fluoridone, but not necessarily to 6 parts per billion
Okay.
fluoridone. It's a little trippy. And like I said, I mean, I think I alluded to earlier, some of my weed science colleagues, like, terrestrial agricultural weed scientists, that might me saying that might give them the cringes a little bit, but thinking about the realities of how herbicides are used for aquatic plant management, that's just sort of a verifiable reality that using at these low doses, you're going to experience a wider range of variation in responses, but that variation comes from the composition, the genetic composition of the plants, and it has nothing to do with the actual location, other than that if the location falls inside one state or the other, you're going to use it at one rate or another.
Okay. Yeah, thank you. Uh, just looking at the chat, I see Ashley did add her email address. So if anybody has follow-up questions, they can reach out to her. We also both dropped in the milfoil mapper link. There's a link to the Thum lab. So definitely scroll through the chat before we finish today if you want any of that information. Any final comments from either of you before I finish up?
Not from me, other than thanks for spending some time with us and letting me spout a bunch of words and slides at you for almost an hour.
It's a great presentation. We really appreciate it. I just want to invite everybody. We have a different topic for our next webinar. We're going to be talking about invasive grasses, both some common ones, things like Phragmites that you probably know quite a bit about. We're also going to be talking about some that you may not be as familiar with, more early detection species. So that'll be on September 25th, and the registration link is live on our presentation page of the MIPIN website, so MIPIN.org. So hopefully you'll join us for that. This was recorded, so if you missed any or you want to share it with anyone, we'll get the closed captioning put in there, and it should be up sometime before the end of the year. So I will definitely email everybody who's registered today and let you know when that's available. And any other questions you have, you can direct to me or to any of our speakers today. So thank you all for being with us.
Thank you.
Have a good day.